Hi everyone, I'm Xiaoyue from CSBL. Today, my presentation topic for Journal Club is the surface-localized CsgF condensate. It's a gatekeeper in bacterial curil subunit secretion, which published on Nature Communications last year. Here are the highlights of this journal. In this study, researchers demonstrated that CsgF phase separates in vitro and that the ability of CsgF variants to phase separate is tightly correlated with the CsgF function during curil biogenesis. Substitution of phenylalanine residues in the CsgF N-terminus both reduces the propensity of CsgF to phase separate and impair the curil assembly. Exogenesis addition of purified CsgF complemented CsgF knockout cells. And CsgF on the cell surface modulated the secretion of CsgA, the curil major subunit to the cell surface. And they also proposed that multi-component CsgF and B condensates from a nucleation-competent complex that templates CsgA amyloid formation on the cell surface. Here I will briefly introduce about the protein secretion systems in bacteria. In gram-negative bacteria, secretion involves translocation across inner and outer membranes. There are now 6 general classes of protein secretion systems. And gram-positive bacteria share some of the same secretion systems as gram-negative bacteria and also display one system specific to that group, the type 7 system. Curil are the product of a dedicated secretion assembly pathway known as the nucleation precipitation pathway or type A secretion system. Curil fibers are functional amyloids found in the extracellular matrix of enteric bacteria such as E. coli and Salmonella. Curil amyloids are composed of a major subunit called CsgA and a minor subunit called CsgB. CsgA amyloid formation at the cell surface is initiated by the CsgB nucleated protein. 5 accessory proteins are necessary to regulate the secretion of curil subunits and to ensure the proper formation of curil on the cell surface. CsgG is a monomeric membrane pore that spans the outer membrane and serves as the channel for the secretion of CsgA, B, and F. The binding of CsgE to the periplasmic region of CsgG is essential for substrate selection. CsgC is a chaperone-like protein that maintains CsgA in a protease-sensitive state in the periplasmic region of bacterial cells. CsgA is secreted to the outer membrane in a soluble unpolymerized form. On the cell surface, CsgB nucleates CsgA amyloid formation. The accessory protein CsgF is essential for the proper anchoring of CsgB onto the cell surface, and without the CsgF, curil formation is disrupted. Recent structural studies using cryo-EM have revealed that the N-terminus of CsgF arranges on the extracellular part of CsgG. In this study, researchers demonstrate that CsgF readily phase separates in vitro and that aromatic residues in the N-terminal region of CsgF are critical for both its phase separation activity in vitro and curil biogenesis in vivo. So their study focused on the interaction between CsgF and the curil subunits during the formation of functional amyloids in bacteria. Firstly, researchers analyzed the structure of CsgF. Mature CsgF has 119 amino acids. Animal studies on CsgF have shown that both the N-terminus and C-terminus are unstructured, while the middle region is a mixture of alpha helices and beta strands. Additionally, CsgF is rich in asparagine and glutamine residues, which is one of the characteristics of prion proteins. Prions are proteins that can switch from non-aggregated states to self-templating highly ordered aggregates. As with low complexity domains and intrinsically disordered regions undergo phase separation. Therefore, they subjected wild-type CsgF to prion-like amino acid composition analysis. PLAAC predicted that the N-terminus of CsgF is prion-like as shown in Figure C. The researchers also validated phase separation of CsgF by observing turbidity fluorescence imaging and FRAP measurements. Since NMR has shown that both N-termini and C-termini are unstructured, they made N and C-terminal truncation mutants of CsgF to determine if either IDR was necessary for its phase separation activity as shown in Figure A. And as shown in Figure B, the turbidity of C-terminal truncation samples increased as the protein concentration increased. Similar with wild-type CsgF, and the condensate formation also can be observed as shown in Figure C. However, C-terminal truncation did not show concentration-dependent turbidity changes or visible droplet formation. These proteins were centrifuged and the supernatant and the palette fractions were subjected to STS page to quantify proteins in the dilute and the condensed phase. The amount of C-terminal truncation in the palette fractions was reduced by about 20% relative to wild-type, while most of the N-terminal truncation remained in the supernatant. It indicates that the C-terminus appears to be necessary for the phase separation activity of wild-type CsgF rather than C-terminus. Studies have demonstrated that aromatic residues in IDRs determine the phase behavior of proteins. Wild-type CsgF has a total of 7 phenylalanine and 3 tyrosine residues, with 4 phenylalanine residues and 1 tyrosine residues in N-terminal region. They created serine substitutions of phenylalanine residues located in the CsgF N-terminus and measured how the phase separation activity of wild-type CsgF varies with changes to the number of phenylalanine residues. Firstly, turbidity measurements shown in Figure H indicated that as the number of phenylalanine residues decreased, there was a corresponding reduction in wild-type CsgF phase separation, and the same result showed in sedimentation assay and the DIC images. This suggested that the phenylalanine residues in the N-terminus are required to promote phase separation of wild-type CsgF. CsgF assists CsGP in forming a functional nucleated complex on the bacterial surface. In the absence of CsgF, cells fail to assemble as much curil on the cell surface as wild-type. To understand the role of N-terminal and C-terminal IDRs of CsgF, they assayed the ability of N and C-terminal truncation to complement wild-type CsgF function in vivo. Curil-producing wild-type cell harboring empty vector can stain red on Congo red indicator plates. A CsgF knockout cell harboring empty vector exhibit a pink color phenotype. Then remove the cells from CR plates after 28 hours of growth. CsgF knockout cells underlying agar was red, because secreted curil subunits that assemble into CR binding polymers but did not attach to the cells. The CsgF knockout strain was complemented by expressing full size of CsgF with plasmid, but when expressing N-terminal truncated CsgF, the phenotype matches the CsgF knockout strain. Additionally, when expressing C-terminal truncated CsgF, remained white even after 28-hour growth. So failure of C and N-terminal truncated CsgF expression to complement CsgF knockout cells indicated that both N-term and C-terminal regions of CsgF are necessary for the function of wild-type CsgF. To better understand how the N and the C terminal domains contribute to the Y-type CsgF function in vivo, CsgF knockout strains harboring N-term and C-terminal truncated CsgF plasmid were characterized. CsgA that is secreted by coliforming cells is assembled as amyloid fibers on the outer membrane. So they measured the CsgA by SDS page in whole cell and agar plug to conclude that the CsgF knockout strains CsgA fails to polymerize on the cell surface, allowing unpolymerized CsgA to secrete away from the cell surface. Similarly, CsgA knockout cells with C-terminal and N-terminal truncated plasmid were not detected in the whole cell but were found in the agar plugs. Therefore, CsgF showed similar result with figure A. So these results indicates that both the N-terminal and the C-terminal regions are required for the proper function of CsgF on the outer membrane, and without the N-terminal region, CsgF failed to secrete to the cell surface. Next, researchers focused on the ability of phenylalanine mutants to complement CsgF knockout cells, and as shown in figure A and B, none of the phenylalanine mutants complemented CsgF knockout cells, and a single mutation was enough to impair the function of Y-type CsgF and the curil assembly. A single phenylalanine mutation can also affect the phase separation propensity of Y-type CsgF. Previous study reported that the unpolymerized soluble CsgA that is secreted away from a CsgB knockout strain can assemble on the surface of CsgF knockout strain by the process of interbacterial complementation. And the CsgF-B double mutant cells are better CsgA donors compared to CsgB single mutant cells. So in this study, researchers performed a CRSA on CsgA donors along with the CsgF knockout and its variants. And to see the presence of CsgF on the bacterial surface, they carried out intact cell double assays. For CsgA secretion, they observed the differences in the amount of CsgA in the plug for these non-curil producing cells on SDS page. Together with the immunofluorescence assays, the result indicated that Y-type CsgF localizes to the bacterial surface and is a regulator of CsgA secretion in the non-curil producing cells. Next, researchers validated whether CsgB could interact with the Y-type CsgF condensates. First, when CsgF condensates were added to the CsgB, both proteins colocalized. Then, to elucidate the nature of CsgB in the phase-separated droplets of Y-type CsgF, they carried out FRAP measurements. An SDS page was also performed to further understand the nature of CsgB in the presence of Y-type CsgF. All of results indicate that the CsgB interact with Y-type CsgF droplet and that CsgB undergoes amyloid transition with the dynamic CsgF condensates. So in this study, researchers demonstrate that the curil accessory protein CsgF undergoes phase separation. Phenylalanine residues in the N-terminal region are stickers responsible for the assembly of CsgF condensates. And both the N-terminal and C-terminal regions are required for the proper function of CsgF on the outer membrane, and the CsgF failed to secrete to the cell surface without the N-terminal region. Exogenously added CsgF protein complemented CsgF knockout cells, and CsgF condensates provided a platform to promote the aggregation of CsgB. CsgF assembled on the outer membrane regulates the secretion of CsgA. Taken together, this study provides insights into the interplay of multi-component CsgF condensate and its influence on curil amyloid biogenesis. This is the end of my presentation. Thank you for your listening.